Hello friends, today I'll be discussing with the topic sequence analysis tool. Here I'm going to introduce about some tools which can be used to analyze, to study the sequences. When I say sequences, it will be very clear. We are speaking about the nucleotide sequences and the amino acid sequences. So, sequence analysis tool. Before we move on with the different sequence analysis tools, it should be very clear for what we are using this. Because we know that all the life in the world exists based on the beautiful arrangement of the nucleotides, followed by the beautiful arrangement of amino acid sequences in the protein. How they are being expressed, which determines how the characters are formed. So to understand this sequence, it's very important. So sequence analysis is the process which helps us to identify the sequences that encodes the protein and to discover how these sequences are being expressed and how they bring about their uh, cellular process. So it provides information about gene expression and protein expression. So there are various bioinformatic tools available for nucleotide, amino acid and analysis. Many of them are freely available, some we can download it. And the examples are ORF Finder, BLAST, FASTA, BITS, CDAT, etc. So here I'll be just introducing about the BLAST and the FASTA. We'll start with BLAST. BLAST, the full form is Basic Local Alignment Search Tool. Basic Local Alignment Search Tool. And this is a tool which I use for comparing primary biological sequences such as amino acid sequences and the nucleotide sequences of the DNA. And as I said, this we can simply search through www in the interface, or it can be downloaded by the extension ncbi.nlm.nih.gov/blast from the NCBI portal. And different type of BLAST programs are available according to the purpose what the researcher uh, have. Like for example, BLAST N stands for, it is a program, it's a BLAST, that is where I'm going to search for the perfect alignment, perfect match of my query sequence. If I'm having a sequence, just imagine if I'm having a sequence, I'm having a DNA sequence or a protein sequence. I want to find this the comparison or the I want to find its link with the already present sequences in the database. So there are so many programs, and whether it is a nucleotide sequence, whether it is a protein sequence, accordingly, what is my purpose? Accordingly, there are different programs available. So BLAST-N. So this stands for N stands for nucleotide query sequence. So I'm having a nucleotide query sequence, and I'll be comparing it with a sequence, nucleotide sequence itself, a DNA sequence which are already present in the database. Second one is BLAST P. So here the query sequence changes. Here the query sequence is a protein, and I'll be comparing it with the protein sequence of a database. Then next one is BLAST X. Here the query sequence I'm having is a translated nucleotide query sequence. That is the result what I get from a DNA. That translated sequence I'll be comparing with the protein sequence databases, because after translation it will be almost like a protein itself. So I'll be comparing it with already present protein sequences. Next, I could see that it is BLAST. It is the BLAST T-BLAST-N. Actually, it should be T-BLAST-N. which is a protein query sequence against translated nucleotide sequence databases. Next, we'll be having tBLAST-X. It is a translated nucleotide query sequence against translated nucleotide database. Next program is PSI BLAST, which finds distant relatives of a protein. Next is the MEGABLAST, which is faster program, which is used for large number of input sequences which are being compared with already present database. So as I said, there are different types of programs in BLAST, and these are being used based on the query sequence input, the database where I am going to compare it. Like sometimes I may be using the collection of nucleotide sequence come. Sometimes I'll be comparing it with the protein database, or sometimes I'll be comparing it with the translated results, or sometimes the nucleotide sequences which are to be translated likewise. So what is being compared? That is also important. And here I could say that BLAST is more effective for protein sequences than DNA sequences. Next is the FASTA. FASTA stands for fast all, which pronounced as FASTA. It is not FASTA, actually FASTA, and it stands for fast all. And it's a we can say here it is a one of the popularly used DNA and protein sequence alignment database scanning program. Here also we are having a query sequence and we are searching the best match in the already present database. And this was created in 1988 by W.R. Pearson and D.J. Lippmann. And as I said, it is used to search similarity between query sequences and any group of sequences of the same type. That is, if I'll be finding if I'm having a query sequence as nucleotide, I'll be comparing it with the nucleotide or protein sequences in the database. And here the FASTA look for the best word matching or sequence pattern. Like it will be having a set. It looks for the like us. It will not simply stand for a single nucleotide. Like it goes for matching word like K, like a length of 15 base pairs or 15 amino acid sequence. It will be comparing it with that much of the sequences in the database, and then it build a local alignment. So if I'm taking like a 12k or 12 base pair I'm taking, so what it do is it will be comparing that much of length, and the best match will be taken, and then alignment will be done. We'll be discussing it in the next video. And here also it can be searched in the web, or we can download it also. So there are different programs available. So we'll just move on with different programs. That is FASTA. It compares it is the normal one which compares the query sequences and a group of sequences of the same type. If I'm comparing a query nucleotide, I'll be comparing it with the database already present. Next is FAST-Hex. It compares a translated nucleotide, query sequences, and a group of protein sequences. FAST-Y. It compares a DNA sequence to a protein sequence database. FAST-S. It compares set of short peptide fragments against protein database. So that was the two different alignment sequences available. So thank you.